So our research focuses on inter-organ communications, and especially in the context of ischemic heart disease. And initially, we start with the communications through cells, focusing on the bone marrow and the heart. And we have revealed that a number of interactions between the stem cells and the bone marrow microenvironment that impact on the stem cell mobilization into the ischemic heart. We have also worked on how once the cells are recruited in the ischemic heart, how they interact with the damaged environment to exit the bioactivities of these cells, including the repair. So recently we found that exosomes seems to be a very important player in mediating these communications, such as Spanish paper show that. So here I'm going to share our experience of learning in this field in the last few years. And as you all know, there are many different kinds of vesicles. Now this varies directly but through the cytoplasmic membrane, and all this called exosomes generated through the endosome and multivascular body, and then fuse and release this kind of intravascular vesicles that seems to be very particularly unique in their biogenesis pathway and perhaps in the biological function, because that looks like the biogenesis is different, so seems to be reflect the biological activities of or the functional states of the cells. So, but it's very difficult to study in this field, as you can see here. That the ones we isolate the cells, and these particles, their size is like that, exosomes, and no quite overlap with different other vesicles, also non-vascular particles. And also density is also kind of overlap with a lot of particles in our body fluid. So especially in the plasma, the total other type of particles may be 10,000 times more abundant than the exosomes. So this raises a lot of challenges for us to study in this field. And also, just to give an overview of this is the size of exosomes versus the nuclear, and then this is the platelet. So different size of molecules give some impression how big is that. Looks like this is the nuclear, and this is the exosome. So just for information. So we learned that in this field, there are quite a lot of physiological roles that are not quite understood. Even though the exosomes are produced by almost every cells, even from the very old ancient archaeotype bacteria and also plants. So it's quite some conserved pathway of regeneration. For example, it's produced through initiated through this endocytosis pathway. So as we know that now the receptor-mediated signaling in the major part, perhaps most of them actually involve in the endocytosis pathway. So basically in that we think this generation pathway is relates to the overall different type of cells signaling state, so functional state and signaling. And also it's not quite clear about this sorting, how this inward budding of this in the formation of this future exosomes, that how these molecules are are sorted into these small particles. It's quite some RNA binding proteins and also different microRNA binding proteins that looks like how the specificity is determined or through, rather non-specific mechanism, how the combination comes together to determine the specific packing. So I'm going to come back again in a little while. And also that, once they have a multivascular body, so they have two choices to make, or go to an exosome to degrade it, or go traffic to the membrane, dock on the membrane to release. This is either go this way or go that way, and I think this is very highly dependent on the metabolic states of the cells. I thought this is quite and a lot of process in there. And once they secreted, whether how whether they stay in the local, how they pass. Exosomes is very famous for pass through the brain-blood barrier and how they do it. There's no clear evidence from my understanding at this point how they do it. And also, lastly, that how they target the target tissues and how they escape the lysosome degradation to exert the biological effect of these biological cargoes. I think also not very quite clear. And if so, here there's an example. Here is the structure of the exosomes, and from this recent paper published in MD Anderson, it looks like they compare 14 types of different cells, showing that even the biomarkers are kind of homogeneous, and also that about 40% of all the proteins are shared by all types of exosomes. So this raises the question whether how the specificity determined by the proteins or by different other mechanisms, or so also the sorting, and so there are clearly some changes in the lipid structure in the membrane, and as I just alluded to, the proteins, RNA binding proteins, mRNAs, and different type of mRNAs get packed into this one. What's the signaling aspect involved in this? I think is totally quite some understanding, but still largely unknown. Another one is the microRNA is a very short stretch of, especially here in this, inside the exosome is a very single-stranded RNA. So, so this is a group from Harvard, from Ron Kahn's lab. They analyze, compare the 6 cell lines of different microRNAs, how these microRNAs are packed into these different cell lines. They compare, they find some RNAs with a certain motif and can selectively highly enriched in all the exosomes. So it kind of come up with the idea that whether in addition to the RNA binding protein, is that kind of or lipid component, whether also the RNA itself, also even though it's a very small short stretch of RNA, also involved in the selectivity of the packing into the exosomes. And lastly, I think that the biodistribution, so in this paper, they summarize quite a few years of all the work that people use to inject the exosome into the blood, then look at the biodistributions. So they exhausted all the publications by that point. So basically, they find that in the blood, after injection, 2 minutes is just 30% remaining. 30 minutes, almost all gone, and 1 hour, so it's gone. So there's a lot of questions whether, the targeting, how you expect, and where they go, kind of question. So they also further analyze that. They analyzed these papers. They found that, looks like quite a lot in the liver, and some in the lung, also, but in the heart seems to be not much, and doesn't seem to be very encouraging for the heart. So as you all know, that now this exosome as a natural product secreted by live cells that people are engineering these exosomes to as a biological carrier for therapeutic delivery. And you can see that, different modality I'm not going to talk about. So a lot of enthusiasm. So we are really concerned about whether they go to heart or not go to the heart. So this is more concern to us. So we developed this strategy really to look at the cardiac the cell-specific exosome proteins where they go. So we developed this transgenic mice where in the mRNA to protein translation, they have this mutation. They incorporate this particular amino acid into the newly synthesized protein instead of methionine. So it's kind of a label. This label can be picked up by chemical reaction. So we found that the labeling is very efficient and specific, doesn't affect the exosomes, and we found that we can pretty much very excitingly identify this RNA binding on this protein exosome-derived proteins by this green color in different type of cells. So it looks still quite a lot of exosome proteins got into the heart, and also that we can compare, and we shown that especially that after ischemic injury and the exosome protein got much more in the heart. So it's the injury. Now we are developing because of this labeling of exosome protein. So we are developing this affinity method, kind of clinical reaction based method to isolate the tissue specific exosomes that have some preliminary access. So our approach for the tissue specific exosomes, one is in the mice through this transgenic mice. So produce this clinical reaction based isolation of exosome specific proteins. As we all know, just introduced, is not that abundant. Tissue specific molecules on the surface of any exosome from any cells are not that abundant. So basically in that we are still can be done, but it is a little bit challenge. And also we use CD63 on a tagging. We also use do some a lot of membrane proteomics to identify different tissue membranes to extrapolate whether these proteins also exist on the exosomes coming from these cells. So kind of compare different cell type derived exosomes for chromatins that to identify. We have identified some proteins in the cardiomyocyte, which a few of them mostly are ion channels. So also, I'm not sure how many minutes we have. So just to introduce one more study we have performed, actually just already published. I introduced the protein synthesis translation metabolic labeling just before. Now we also use the same strategy to label RNA metabolically and through this UPRT. So RNA normally do not incorporate this 4-thiol uracil, but by the transgenic expression of this one, so they're able to incorporate the 4-TU into the newly synthesized RNA. So these RNAs can be picked up by clinical reaction or by SNAP sequencing. So we were able to identify the tissue-specific RNA from the exosomes. And from this particular study, we shown that after myocardial infarction, we found that because only the cardiomyocyte express this transgene, so cardiomyocyte mRNA is enabled that after a certain period of time, after myocardial infarction, very quickly, 6 hours, we find that these cardiac-derived exosomes are very efficiently identified in the circulation RNA out of the total amount of exosomes. And also we found that out of many different organs, the lung express a lot of this RNA from cardiac labeled microRNA in the lung, and also particularly in the lung endothelial cells, also in the epithelial and the bone marrow derived lung cells. So we don't know why it's overall overwhelmingly go to the lung, but because this is very important molecules in as you from publication from Eric Olsen and many different groups from very high impact journals showing that they have a very strong functional impact in the body, even though it's a cardiac specific microRNA involving the metabolic regulation of total body. Okay, so I think my time's up. And basically, I want to show you is that through this study, we were able to show that by this RNA-specific labeling of exosome from cardiomyocyte, we were able to identify in the basal level there is a transfer of microRNA 208 from the heart into the lung, and predominantly regulate is targeted in the lung, and actually this targeting of this downstream gene have an impact in the NF-kappa B pathway, basically increase the inflammation in the lung. So looks like there are short transient myocardial ischemia injury inducing this microRNA transfer through the exosomes that be able to induce also transient inflammation in the lung or increase a little bit of the membrane thickness and the edema. So with that, I'm going to introduce, thank the investigators in my lab, just moved from UAB to SASTAC and established my new teams, and we are recruiting, so welcome, and every teacher's place refer and us, and we are recruiting for students, the postdoc researchers. Thank you very much.